Okay, so it's great to have this meeting in San Diego, so we need to travel. And thanks to organizers, especially the guy for taking the main hit with this meeting. So I'm going to talk about sleep, and I think it's a perfect time, right? Late afternoon for those who travel from Europe, especially. So this is a work supported by CRCNS grant we have with Bruce McNaughton at UC Irvine, and it's pretty much at the end, at NC phase. We have some number of interesting results, but I think this is the most kind of exciting from my perspective. We have a bioarchive paper, and it's under review, so I guess if somebody interested, you can find more details online. So, oops, but it doesn't check it. So, should I make a full screen? Or no, it's full screen, right? Okay. Yeah, again, so this sort of topic of my talk about sleep and memory consolidation, if in my opinion, one maybe more striking kind of argument in support of ideas that sleep playing big role in memory consolidation is how stereotype and how kind of constrained sleep structure across species. So here on the top left, you see typical, I didn't make it screen right, typical human hypnogram, which sort of showing characteristic transitions between slow wave sleep and REM sleep. Below are recordings from fly, where at least those are claiming that there are two phases of sleep, kind of high neural activity representing something equivalent to paradoxical sleep, which cannot really be called REM sleep in flies, and then phases of something equivalent to slow wave sleep. Now, the most basic idea behind the role of sleep and memory consolidation illustrated on this right side. If neuron fire in certain order, when animal or human doing certain tasks, it's kind of change synapses between them. And then during sleep, the same pattern can be reactivated, possibly at some accelerated rate. And this is kind of one fundamental idea behind the role of sleep and memory consolidation, idea of replay or reactivation. So another kind of making a step forward, another great idea in this field was the idea of complementary learning systems, which sort of are almost 30 years old, and it was proposed in different flavors from different studies, but essentially idea that when we learn something, specifically declarative memories, we first learn them in sort of fast learning, most parts representing the hippocampal network, and then over time, over multiple periods of night sleep, they map to the cortex. And there are at least two, more than two, but two which I will be emphasizing, two players in this game, sort of hippocampal chakra ripples, which encode those sequences, which I showed in previous slide, and then slow oscillations, which make cortex kind of ready to accept that information. So it's definitely help, and I guess a very basic idea behind why this kind of dual memory system exists is because it's create an offline period when new memories can be encoded to the cortex with less chances to damage what already is there, because that's kind of the problem, and we know it's problem in machine learning, catastrophic forgetting, and so on and so on. However, in principle, this idea alone not really solving the problem. This is kind of real cartoon. Let's say we have some memory represented by those three synapses between four cells, and then the hippocampus trying to teach cortex something new. And in principle, if there is direct conflict between sort of order spiking, which neurons are involved between those two memories, it's possible to erase that memory. And in fact, I wouldn't have time to show it, but we can show it in the model that if we have some memory phase encoded to the cortex, and we just trying to write over something encoded by the hippocampal ripples, it will destroy the old memory. So again, this problem known artificial neural networks, and it has been shown one basic idea coming from neuroscience, in fact, how to sort of mitigate the problem is the idea of precursor. Essentially, if we can somehow interleave new training with old data, we can avoid the catastrophic forgetting. It has been, again, shown in machine learning, and now the brain obviously does not have access to all samples in an explicit way. However, it keeps sort of memory traces of all, synaptic traces of all memories. So the question is, can it use this information to sort of do something equivalent to rehearsal, but without explicit access to all samples? Okay, so to address this question, we use two tools. One is the biophysical model, which, I mean, the basic foundation of it was developed almost 10 years ago. And what is nice about this model, it can kind of integrate lots of pieces which have been developed by many labs, models of spindles, slow waves, gamma oscillation. And essentially, it's combined them together in a way that we can model different sleep phases. Like what is shown on top is wake state, then we go to stage 2 with a bunch of spindles, then we go to slow waves, then we have sample REM sleep, and back to wake. So it's all organized by sort of changes in some neuromodulatory tone in the model. On experimental side, work done in the Sensorimotor lab involve this mouse sitting in sort of virtual reality environment on a wheel, and it can be exposed to two different visual environments which can be trained, and we can switch one environment by another, and sort of we use this to essentially create a notion of familiar memory which could be trained first, and then some new memory which can be competing with old one to try to address again the question how it's all working. Now, finally, last piece, what we call in the model memories essentially the sequences of activating neurons. This is from paper we published years ago, and we see the sequence ABCD, and in this specific case, we train it by activating neurons in order B, A, C, D, etc. And you see next, we tested by activating just the first letter B, and it's kind of replayed, but performance on the right, it says before sleep is kind of low. And after sleep, it's greatly enhanced because of reactivation. Again, that has been shown before. So now the protocol we use in this new study involves two memories, and we use simple case of linear memories. So first we train this kind of ABCDDA memory trace in a way, and we take it strong enough, it shows good performance. We can add memory, we can add sleep after to kind of consolidate it. But importantly, now we bring the second memory sequence, which is ABCD, which kind of opposite, involves same group of neurons but activated in the opposite order. And we try to encode it using idea of how hippocampal ripples interact with cortical slow waves. The protocol shown on top, we test, we train this familiar memory, we test it again, we now have slow wave sleep, when we try to index and code a new one, and we test it after the sleep. What we also can do, we can try to bring this hippocampal ripple at different phases of slow oscillation. So again, slow waves, it's up and down states, and up state lasts for 3, 4, 5, 600 milliseconds. So so ripples generally generated at different times. There are some evidence showing that they biased at down to up transition of slow waves, and more recently work from Buzaki lab shows it's also maybe peak at up to down transition. And essentially we can play the model, we can apply ripple at different times, and we can see what it can do to the memory. So this is what we got. So going from left to right. So we have a baseline, we have two memories again. One is S1 is a familiar memory, and blue is S2 is a new memory. So in baseline, they show some low performance, again, we can test them. Then we train the first memory, which is the familiar one. You see the kind of red bar. This red bar goes up, and the blue bar is actually goes down a bit because they directly compete for resources. Now we have a period of indexing of new memory during sleep. And you can see that we kind of having great outcome. So old memory, familiar memory actually improved performance slightly. And the new memory, blue one, gets in a great gain in performance. So the both of them sort of improves. Now, what happens if we try to index new memory in the middle of upstate? And again, first two kind of group of bars are the same, but the third one now shows very different result. We try to index in the middle and it doesn't drop. So the blue bar is still very low. However, the red one, which shows all familiar memory, now increase almost 100%. So despite our efforts to kind of index new memory, Cortex didn't respond and instead sort of keep improving the old one. And now if we do indexing at the end, at the up to down transition, we again kind of get the result somewhat similar to the first case. So we still have a gain in the old memory and we train to some degree new one, just less much than in down to up indexing. Okay, so what happens? What we can do in the model, we can look directly on reactivation replay, and we can do it in kind of a bit complex way. So we can, first of all, we can measure reactivation of each of those two memories. And we can also check it at different phases of upstate. So we can basically, we can separate upstate in two kind of phases. Time when we index new memory and time when we don't. And we can check what happens with the reactivation of both old and new memories at each of those times. Like for example, on the top, we index near down to up transition. So we can look at that time which memory S1 or S2 is reactivated. And we can look what happens during subsequent part of upstate when no indexing. We can repeat the same when we do it in the middle or in the end. And so what is shown on the right is kind of synaptic connectivity, but what is shown not the weights, but amount of reactivation. So if it's each dot here would represent the synapse between two neurons, and if there is no color, meaning that reactivation is below some threshold. And if there is a color, meaning that reactivation is higher than threshold, and you kind of, because we have a simple case of two linear sequences, depending where they are, where dot is on diagonal above or below, it really will support one memory or another. So everything below in those kind of red boxes would support old memory, and everything above in blue boxes or round would support a new memory. So this is a case of down to up indexing, and what you can see is that during index phase, we have reactivation of new S2 memory as we would expect. Interestingly, during not index phase, if you look later during upstate, we actually have very nice reactivation of the old memory. So both of them co-reactivated, which is kind of what rehearsal does if you go back to that idea I mentioned. Now, what happens if you try to index them during middle phase of upstate? Again, during index phase, well, so in this case, so S1 is old memory. So we can see that if you try to index in the middle kind of upstate, in both cases, we reactivate old memory. So on the left plot on top, we see the new memory is kind of trying to be reactivated. It's propagated a little bit, but then essentially it fades. So when we try to index cortical upstate by using ripples arriving in the middle, we essentially fail to engage reactivation of the new memory. And if you go to up to down transition, we again see the sort of picture equivalent to the first one, when we have during index phase reactivation of new and during other times reactivation of the old memory. So this is kind of summary. So on the right, we see during not index phase, we always have reactivation of the familiar memory. And during index phase on the left, we can see reactivation of new memories as high blue bars, but only if we pick up right timing for indexing. Now, going to date again, the story behind kind of interesting. We had the results, and it was nice and exciting. And then Ryan, Job, and my lab start talking to just a Hannah and Mark in Bruce McNaughton's lab, and they decided to look at kind of recent data collected for another experiment. But it had two environments. So one was kind of familiar environment, so Amber was exploring it for a while, and then the new environment was presented animal just briefly, was exploring it, and then recordings have been done during both exploration and during subsequent sleep. So what is shown here is a reactivation of old familiar and new memory, now experimental data, and they don't have obvious access to synaptic waves, but they can do simply generated sort of during exploration phase, and try to match the template to what happens during sleep. And it can be done also in the same fashion. So each upstate can be divided into beginning, middle, and end phase, and we can look what happens, which memory gets activated at each time. If you look at the middle plot, so middle phase of upstate almost always reactivate red, which is familiar memory. So we don't have access to when ripples are arriving, but essentially it suggests that if they arrive or not, cortex will reactivate again familiar memory in the middle of upstate, and during sort of first and last third of upstate, kind of corresponding down to up to down transition, when I was talking about model, it was reactivated both old and new memory. And I should say that because obviously in some cases upstate can happen without ripples in vivo, experimental data will be more biased to reactivation of familiar one versus model where we can exactly control what we do. So this is data from two mouses. We see again middle phase reactivate red familiar memory, and beginning and end of upstate reactivate kind of mixed of old and new memories. And this is if you kind of process it's modern data slightly different, this is what model predicts. Pretty much identical pictures. So we have a strong reactivation of largely familiar memory in the middle of upstate, and mixed activation during other phases. This is my kind of concluding slide. So basically, what we're suggesting is that if there is a familiar memory, this top kind of red cartoon encoded to the cortical network, and we're trying to embed a new memory. So first, will we succeed or not depends on when we apply that embedded, when check will ripple arrive from the cortex. So if it's arrive at the beginning or end of upstate, it does reactivate, but at the same time, the rest of the upstate will reactivate old memory, familiar memory. So we have perfect example of, again, this recursal, and both memories get embedded. If it's arrived in the middle, it's not triggering really any reactivation, and we done with keeping old memory. So this is my conclusions, which got it not letting me to speak out, but essentially I already said it. So what we propose is that brain use the recursal strategy, but again in a way, kind of smart way. There is no access to all samples, but it can reactivate all the new memory in a relieved way to avoid forgetting. And this work was supported again by CRCNS, and it has some previous history supported by other agencies, and Ryan and Rajat with all this local stuff. Thank you.
Could you share your I just wanted to see that second thing. Questions?
Yeah, interesting work. I was wondering about the up-down transition stimulation. I mean, I guess because doesn't it then fall on a sort of inactive state, or are you saying right before there would be a transition, or is it just somehow prolonging the upstate? I guess.
Yeah, it's a great question, and we actually, in fact, interestingly, when we built the model, we predicted that up to down will work, middle not, but we didn't look to down to up. And then when Rajat looked at experimental data, they found that down to up actually shows lots of reactivation of, again, new memory phase. We looked at the model back and we found it as well. So I think what you said is right. First, it does prolong it. We had some paper 2 years ago, and when the ripple arrives at the end, and basically it arrives not when it's already falling down. It arrives, I don't know the exact timing, but it's when activity close to end, like 100 milliseconds before something. And it does extend it. Why it's engaged in new reactivation and cannot do it in the middle, we're less sure. I guess one guess is activity kind of slowing down towards the end of upstate, and probably easier to engage cortical network to fire in certain order versus middle when it's heavily engaged zone activity, but it's a bit open question.